I'd like to introduce our speaker today. This is John Carroll. He got his PhD here at SoMAS. He actually got his undergraduate at Southampton College. And then he went off and worked down in Florida Bay for the USGS, came back and did his PhD here at SoMAS. After that, he went to UNCW, did a postdoc there. While he was here, he was doing a lot of base scallop work and kind of predator-prey dynamics on them. The only ecology paper that I have, John published, looking at the edge of seagrass as an ecological trap. And then he started working with oysters and working with kind of this boring sponges in oyster and thinking about issues of changing pH and how that might give the advantage to the sponge over the oyster. And then he went from there to Georgia Southern, where he's now the director for the Institute of Coastal Plain Science. So I'm very excited for John to come back. Make sure, pay attention, please ask very difficult questions, okay?
All right, thank you, Brad, for that introduction, and thank you everybody for coming today. I looked outside on the bulletin board. It looks like you already had an oyster talk earlier this semester, but I promise mine's going to be quite a bit different than that. And I'm going to focus on some of the research that my lab has been doing, ever since I left Stony Brook, looking at controls of oyster populations in coastal plain estuaries. And like Brad mentioned, I did start here, so this is I don't know how the laser pointer works, but this is artificial seagrass mats, and so I maybe got Brad into a little bit of trouble when we were making these mats in the old gymnasium at Southampton College. But I did a lot of work with the base scallop restoration effort while I was here. And I will note that after I left, there started to be problems with scallops again. So I don't know if there's any kind of correlation there or not. But there's no scallops where I moved to North Carolina or seagrass. And there's definitely no seagrass and no scallops in Georgia. And so I've shifted gears and started looking at oysters and the factors that are influencing oysters along our coastlines. And so why should we care about oysters? Well, they form these really extensive reefs where I live, much different, I feel like, than you probably are experiencing up here. And they do that by releasing their gametes into the water. Their larvae float around for about three weeks. When they're ready to settle, they look for hard substrates. Usually that means they're settling on top of other oysters. And as you have these generations of oysters settle on top of each other, they form these complex three-dimensional reef structures. Now, this is anybody that gives an oyster talk is going to show a picture that looks like this, showing the filtration capacity of oysters. This is a tank that has oysters. This is one that doesn't. And so one of the ecosystem services that oysters provide is this filtration capacity. But they provide a lot of other ecosystem services. In particular, they're vital habitat for a lot of our coastal species. These are recreationally important fisheries. These are spots and things. We have our commercially important blue crabs, so they're a commercial product, but they also support communities of other commercially and recreationally important species. And they also protect our shorelines. And so one of the things we're really interested about oysters in Georgia is this idea of shoreline protection. This hard structure baffles energy, prevents sediment resuspension, prevents erosion. And actually, when we build oyster reefs, we see the marsh migrate toward those reefs. Unfortunately, oysters have experienced dramatic declines. The biggest culprit, obviously, is overfishing. And the problem with overfishing is not just removing the oysters, but also removing their habitat. When we're trying to restore oysters, we've inadvertently introduced some diseases. And then with coastal development, we have lots of water quality problems. In Georgia, this is the landings. This is fishery-dependent data. So you have to take it with a little bit of grain of salt. But this is basically the figure you'll see for oyster landings everywhere along the Atlantic coast. And this is just to say that in the past, we had a lot more oysters than we have now. And so in Georgia, what we're interested in doing is restoring and enhancing this oyster resource. We do this with bagged shell materials that we put along shorelines. We do enhancement for a number of reasons. One, to protect the shorelines. Two, to enhance habitat for fisheries. And three, also for the oyster fishery itself. And so there's a couple of different umbrellas within the Georgia DNR that does oyster enhancement. And so I'm interested then in understanding what are some of the factors that control oyster populations so that we could do these sort of enhancement the oyster restoration enhancement more successful. And like all organisms, their population distribution is set by abiotic and biotic variables. Oysters are incredibly well studied. And so we know a lot about the abiotic variables that affect oysters. We know a lot about the biotic variables that affect oysters. And I'm going to tell you two-ish stories today. First, I'll focus on parasites as a biotic control, and then I'll focus on groundwater as an abiotic control of oysters in Georgia. Okay, so parasites. So there are lots of parasites on oyster reefs. Parasites themselves aren't necessarily bad. They're actually critical for ecosystems. But when we're talking about a species we're trying to manage and restore, that's when parasites can become problematic. Like Brad mentioned, I started looking at oyster parasites by looking at this boring sponge from the genus Cliona. And they're not parasites in the true sense in that they don't actually derive nutritive value from their host, but they steal the shell. So they're kleptoparasites. They excavate cavities into the shell. That's where the sponge lives. In real severe infestations, they can penetrate the mantle cavity. You can imagine a filter feeder doesn't like that, and so they have to invest energy into repairing that shell, and that means they have less energy for other things. And so if we look at sponges and if they affect oysters, the answer is yes. And this isn't new. I mean, people have been looking at this since the 1950s, again, because oysters are a commercially valuable product. So people have been concerned about sponges for a long time. So we're not groundbreaking here by saying that the sponges affect the oysters. But when I started working with this, what we found was that we didn't always see that negative effect. And so it suggested that there's some sort of context dependence to this interaction, and it's not maybe necessarily always bad. But I'm hopefully going to convince you that at least in Georgia, it's pretty much always bad. So why do we see variability in these experiments? Well, one of the reasons is obviously we can look at oyster metrics and look at oyster size. Bigger oysters have longer to accumulate the sponge. There's usually larger sponge infestations, so those oysters are having to dedicate more energy to shell repair. Larger oysters also have to put energy into things like reproduction, and so we see the negative effect of sponges being really dramatic on larger oysters than on smaller oysters. So that's intuitive, but we're also interested in what's going on in the environment and how might the environmental conditions be sort of modulating this relationship between oysters and sponges. And so one of the things we're interested in Georgia and throughout most of the Southeast, we only have intertidal oysters. We don't have subtidal oysters. And so we know that the intertidal distribution of oysters can affect oyster demographics. So if we go to our reefs and we look at the low edge of the reef to the high edge, what we see is density of oysters increases with elevation. But we also see some different morphometrics. Those oysters that are in the higher tidal are longer, but they're skinnier. Okay, so they're more densely packed together. They're longer, skinnier oysters. So we know that the sort of location within the tidal prism is going to already impact oyster demographics. So we're interested in how this location might affect this relationship between sponges and oysters. And so if we look at sponges, we see the prevalence also being affected by location in the tidal prism, such that we have higher sponge prevalence in the lower part of the intertidal zone and very low prevalence in the upper part of the zone. And this makes sense because sponges don't like to be exposed to air. And so they're going to be preferring that the place where they're exposed less frequently. But what we also found is that the negative impact of sponges on oysters also changes with tidal elevation. And so in the low intertidal zone, they have a more negative impact on those oysters than they do in the other tidal zones. So we see that the effect of sponge on oysters is impacted by where that relationship is occurring within the tidal prism. We're also interested in things like salinity, because the sponge prefers marine salinities. Oysters can tolerate pretty low salinity compared to other marine organisms, although they do quite well in marine salinity as well. And so we do a sort of salinity gradient here. SS is Savannah State, SK is the Skidaway Institute, and PL is Priest Landing. And so we have this nice salinity gradient where we look at tidal elevation and salinity and the presence of sponge. When we do that, what we see is that all those variables affected the growth rates of oysters. Oysters grew more slowly when they had sponges compared to when they didn't. There's a tidal elevation impact on growth rates, and I could sort of talk to you later about what I think is going on there. But the important thing right here is that site had the biggest effect on growth rates. In fact, the oysters that grew at Savannah State, this is the site with the lowest salinity, even the spongy oysters at Savannah State grew faster than the oysters without sponge at the other two sites. So this is suggesting that site can mitigate maybe some of those negative impacts. Now notice I'm saying site and not salinity, because as it turns out, when you use site as a proxy for salinity, a lot of other things are varying across sites. The triangles here are Savannah State. So Savannah State consistently had more food in the water column. Savannah State had less fouling organisms, so less competitors. These are barnacles and squirts and oyster spat. And so some combination of salinity, food availability, and competition is likely driving this site effect that we saw with this study. Now we're also interested in how the presence of the sponge might impact relationships with other parasites like the pea crab. Anybody that's eaten oysters, you've probably encountered pea crabs. They occur in about 3% of oysters. And so we started to ask whether there's any sort of relationship between these two species, because when I was in North Carolina, it just seemed like the spongy oysters had more of them. And so in fact, when we look at the percent prevalence of pea crabs and oysters that have sponge versus oysters that don't, keeping in mind that the background for pea crabs is about 3% if you look at the literature, what we found consistently, not only in North Carolina, but also in New Jersey and subtidal reefs and in Georgia, that spongy oysters were 2 to 4 times more likely to also have a pea crab. So it suggests that having the sponge may also impact their susceptibility to other parasites. Now, luckily, the impact of both parasites is just additive. So there's not a synergistic interaction happening here. The effect on oyster condition is essentially the same if you just add the negative effects of both parasites. Regardless, the oysters that had both parasites were in poor condition, fairly consistent. And so those are all individual level measurements, but if we look at population level, what we see in Georgia is that our oyster density decreases with increasing sponge prevalence. I mean, I know I'm not explaining a lot of the variability here, but just looking at one variable as potentially driving oysters, I think we're doing a pretty good job. And so again, the more sponge we have, the fewer oysters we have. So we're interested in sort of why. We know we have these sort of negative effects, but why on individuals, but why are we seeing this sort of effect at the population level? And so a colleague of mine that also is a Southampton, Stony Brook alumna, Dr. Amber Stubler, we were deciding to look at the rates of bioerosion that were occurring in our oysters in the Southeast. And we were trying to see if nutrients were an important factor for that. They weren't, so we're going to ignore that. There's 2 bars, but I want you to sort of focus in here, if you can see it. The bioerosion rates what I was measuring in Georgia were an order of magnitude greater than the bioerosion rates that Amber was measuring in North Carolina. And even the bioerosion rates that Amber was measuring in North Carolina were higher than most of the values we could find in the literature. But because my values were so high, Amber assumed I did something wrong. So I repeated the experiment, this time with limestone tiles, and found the same order of magnitude of bioerosion occurring in our Georgia estuaries. And this is intuitive, but as we have more sponge biomass in our substrates, we get higher rates of erosion. This is a table that's not really that important, except that I just want to sort of cue you in on some important stuff down here. In the second experiment, we used limestone tiles, so we knew the surface area of the substrate, and we know the loss rate, and so we can scale that up to kilograms of calcium carbonate per meter squared per year. And in Georgia, the sponges are causing an erosion of about 10 to 20 kilograms of calcium carbonate per meter squared per year. And to put that into perspective, oysters accumulate carbon on reefs on the order of around 20 kilograms of calcium carbonate per meter squared per year. So in Georgia, the oysters, the sponges are potentially causing erosion of anything that the oysters can put out. And when they're more common in that lower intertidal zone, they're really, really erosive. When I moved there, I was told the sponges are aggressive, but now I know what they meant when they said that the sponges were aggressive. So we have to then consider what are the factors that drive the distribution of sponges. And so we have all these surveys along the coast, and we measure a lot of different water quality variables. But the biggest effect on sponge prevalence is just elevation. And so what this tells us is just as long as we put the oysters in the right place, the sponges probably aren't going to be a problem. Now, my lab has started to move into other parasites. And so we're also looking at protozoan parasites, Haplosperdium nelsoni, which causes the MSX disease, Perkensis marinus, which causes dermal disease. And when oysters have these parasites, it usually has a negative effect on their condition. And both of these parasites have been linked to mass mortality events for oysters throughout most of their range on the Atlantic and Gulf coasts. And as it turns out, in sort of pilot studies, when I first moved to Georgia, these parasites are very common. On the order of 80 to sometimes 100% of the oysters that we sample have one of these parasites. If we go to a lot of locations along the coastline, we're getting on average about 90% prevalence of parasites. These are parasites of management concern because they could affect oyster condition and because they've been linked to mass mortality events. So we have a lot of them. So my lab has started to look into what are some of the environmental factors that might be leading to this high parasite prevalence. And so we could do studies. This is the coast of Georgia here. So we can do studies along the coast, and we sort of sample oysters and we look at the parasite loads, and we can sample water quality and just see if there's any sort of environmental parameters that might be driving our parasite prevalence. And when we did this in 2018, we couldn't find any sort of relationships for either parasite and the water quality that we were measuring. But as it turns out, we sample multiple years, and when we did this survey again in 2020, we were able to detect some effects of water quality. So there's variability in parasite prevalence and intensity across years, and when we're able to do a study in 2020 at these 4 sites, so smaller spatial scale but higher resolution water quality data, we can really start to see some of these drivers. In fact, for Haplosperdium, temperature and salinity both had negative effects on prevalence, which is contrary to what's found in the literature. When we look at Perkensis, we actually don't see any relationships with water quality. And so this is also telling us that the drivers of the 2 parasites are going to be different, which is going to create problems for management because you can't sort of make predictions about both parasites using the same water quality parameters. Now, that was just a couple of snapshots at a time. I teamed up with some colleagues at the University of Georgia. We really expanded our spatial extent of our sampling, both along the coast and within creeks. Again, very, very high prevalence of one or both parasites. But now we have more sites, so we have more data, so we can really start to hopefully parse out some of the drivers. And when we do that, this is for Perkensis prevalence. Well, it increases with oyster size. That makes sense because they have more time to accumulate the parasites, but we see a positive relationship with temperature. Interestingly, we see this positive relationship with mud crabs. Not that the mud crabs have anything to do with the parasite, but it lets us know that whatever drives mud crab density, usually things like oyster density, reef rugosity, current flow, might also be driving the presence of this particular parasite. And then we could take our reef location and we could look at landscape scale buffers around the reef. And so we could look at the amount of marsh relative to open water around our reef. And as we increase the amount of marsh around the reef, we see a decrease in this Perkensis prevalence. Now we see the exact opposite patterns with haplosporidium. Again, we see a decrease in haplosporidium prevalence with temperature and salinity. Now this is confirming what we found in Georgia, which again is contrary to what's in most of the literature. But we also see an increase in the prevalence when we increase the marsh to water ratio around the reefs. So again, this is all to say that we have very different potential drivers of these two protozoan parasites. Now, all the studies I just talked about regarding these protozoans were just snapshots in time. We went out in August when we expect the parasite load to be the highest. And so we decided that we needed to increase our temporal resolution of SAM. And so I had a student that went to these 4 sites. This is called Sapelo Island. For reference, this island is the same size as Manhattan, population 60. Okay, so this is a very, very undeveloped island, but it's a National Estuarine Research Reserve. And because of that, it has system-wide monitoring program stations that continuously monitor water quality data every 15 minutes, going back 20 years. And so we can leverage those data sets and pair it with our parasite abundances. We collected oysters every 2 weeks from April through November. And so we know the prevalence at each of those time points. We know the water quality and what it was doing both at the time of collection, but also we can start to look at lags, potential lag time effects of these water quality parameters. And so when we look at this, for Perkensis again, the strongest effect was temperature, but it was actually the minimum temperature 2 months prior to sampling. And so this is letting us know that, we could potentially predict moving forward. If we know it's going to a warm winter, we could predict that we might have a higher prevalence of these parasites, because as the minimum temperature increases in recent past, it leads to an increase in the prevalence of our parasites. We also confirmed that the drivers of Perkensis and haplosporidium are different. This is important for management. But one of the cool things that came out of the haplosporidium work is it wasn't sort of the mean values or the min or max. As it turns out, the biggest driver of haplosporidium presence was the variance in temperature. So more variable temperatures, presumably more stressful for the oysters, leads to higher prevalence of this parasite. Now, I do want to note, as it turns out in Georgia, we're doing all this study with the parasites, but we don't see very, very high intensities. We can detect that they have them, but we're not seeing diseases that are being caused by these parasites. We don't see effects on condition. We don't see mortality events in Georgia. And so it's actually turns out probably these are maybe not very problematic in Georgia at this current moment in time. I think that has to do with the Georgia oysters being better adapted to the other conditions that are usually stressful. When they have these parasites, it's usually a multiple stressor that leads to mortality. The parasites are really proliferate above 20 degrees C. Well, in Georgia, that's like more than half of the year. And so these oysters are probably adapted to what would otherwise be stressful conditions for oysters elsewhere in their range. And maybe that's why we don't see the negative impacts of these parasites. Regardless, how are these biotic interactions affecting management? Well, the boring sponges are having a really negative effect, particularly in Georgia. And I actually think that the presence of boring sponges in Georgia is what's contributing to this intertidal zonation that we find oysters in. We have a lot of these disease-causing parasites, and we could link them now to environmental variables, although they're probably not as important for our oysters in Georgia. And so this is just to say that if you have the proper site selection and you place the oysters in the proper location within the tidal prism, we can mitigate the impacts of these biotic interactions. Okay. Got time. Good. Okay. I'm going to now talk about groundwater discharge as a potential driver. So I'm switching to an abiotic driver of oysters now. And if there are geologists in the room, I'm probably going to get some of this wrong. But I just like to tell this story because I think it's very interesting. So in Georgia, we have very shallow coastal water tables. And this water that makes up the water table could be water of any composition. So initially I thought that it was only going to be fresh water. But our groundwater and our surface water tables here is actually very salty. And it's probably, the water table is probably filling as the high tide is over the marsh, percolating through those sediments, getting into this water table. and then flowing out in this sort of diffuse, you can see the seepage face right here, flowing out to the surface waters again in this sort of diffuse discharge. Now, we don't know what the impact of groundwater on oysters is. It's actually, there's like 3 papers before we started looking at this relationship. And they're all from Texas, where the conditions are quite different than the East Coast. And so we weren't sure, but we know that groundwater can be an important source of nutrients and can stimulate primary productivity, for example. So that may be to the benefit of the oysters. But we know that groundwater can also be a source of contaminants. So maybe that would be to the detriment of the oysters. And we just weren't sure. And so I just want to orient you to where we sampled. We sampled in this sort of meandering creek called Oyster Creek because it has oysters in it. This is Savannah, Georgia. And if you drive on the causeway, a 2-lane road to get out to Tybee Island, Oyster Creek is directly to your south. And I had a colleague in the geology department at Georgia Southern who is a groundwater geologist. And so when she arrived at Georgia Southern, she did sampling in areas along the coast. And so this is her groundwater flux using radon in Oyster Creek. And what you can see is our spatial variability in groundwater flux, like we might expect. And she looked at this map of oyster distribution, and it's really hard to see, but there's thin red bands along some of these shorelines. That's illustrating where the oyster reefs are found in this creek. And she made this observation that, hey, at this area where I have high groundwater, I don't have oysters. And this area where I have low groundwater, we do see oysters. And so we started talking about, is this something worth looking into? And so when I arrived at Georgia Southern, we set up a project to look at this, and then have continued to work in this arena. So this is Jackie Kelly. She's a geologist. This is her water gas diffuser. So the water gets pumped in, the radon goes into gas form. And she can detect it and do all her modeling to sort of make a map of where we see high and low groundwater. And I do all the oyster stuff. So I put out recruitment tiles and growth experiments and oyster demographics. And what we found in Oyster Creek was that we do in fact have the spatial variability. We also have temporal variability. So this is June, July, August, September. And what we find is that we see some months that have high groundwater flux, and we see some months that have low groundwater flux. But regardless of that temporal variability, the areas that have high flux tend to remain high flux areas across time. So then we can sort of see if this spatial variability in groundwater is affecting our oysters. And so if we just go to a bunch of sites, we selected 23 sites in this creek. If we just go to a bunch of sites and say, are there oysters there, yes or no? As it turns out, when we have oysters, we have much lower groundwater flux than when we don't have oysters. Now, this isn't really informative, so we have to look at some of the sort of population scale measurements and some individual responses. And so if we look at density of oysters now, not just presence or absence, and our groundwater flux, and this is relative, what you can see is that at low groundwater flux, we could have the potential for lots of oysters. There's a lot of variability here. So groundwater is obviously not the only thing driving oysters, but we could have a lot of oysters. And as we increase groundwater, we tend to not see oysters, particularly if you just ignore these 2 points here. And so I'm going to just put this arbitrary line. And again, below this, we can have lots of oysters. Above it, we don't really see oysters. So we may be seeing a response of density of our oysters in Oyster Creek due to groundwater flows. So we do a series of experiments. We're like, okay, well, let's look at individual responses to this. And one of the things we do is growth rate. And the scale here is important. What's important is that at relatively high flux rates, where we didn't expect to find oysters, they grow very fast. In fact, our slowest growth was at our site with our lowest groundwater flux. So if we look at the condition of the oysters when we harvest them across the entire range of groundwater flux, it's essentially the same. And so the individual responses that we typically look at for oysters don't seem to be being affected by groundwater, even though it looks like we might have a density effect. So why do we see that density pattern? Well, as it turns out, we also measure recruitment. And these are terracotta tiles that we use for recruitment. These 2 tiles were put out on the same day, collected on the same day a month later, on the same side of the creek, less than 500 meters apart. This is a high groundwater tile. This is a low groundwater tile. This tile has about 4 or 5 oysters on it. This one has over 8. And so we see this potential response in recruitment. Now, we did recruitment monthly, and anybody that works in recruitment of bivalves knows there's a lot of variability across space and time. There's also a lot of variability across space and time with our groundwater. But if we condense those 4 months of data into this one plot, what we see is this negative relationship explaining about 28% of the data between recruitment and groundwater flux. And so just using one variable, we're able to explain about 28% of recruitment in Oyster Creek. And so to me, it suggests that recruitment limitation is occurring in the areas with high groundwater flux, or at least it's an eruption. There's probably larvae there. They're either not undergoing metamorphosis, they might be bopping back up into the water column and moving away, or they might be succumbing to mortality in these areas. And so the question is why? And so this is a big correlation matrix. We have our site characteristics, density, our oyster characteristics, sediment quality, water quality, and then groundwater flux. I want to focus you in on this bottom part. At our high groundwater sites, as groundwater increases, we see strong negative relationships with pH and dissolved oxygen. So groundwater goes up, surface water pH and DO go down. We know that pH has a negative effect on oyster recruitment and larval survival and metamorphosis. And so what this suggests is that it's probably a water quality issue at these high flux sites that are leading to this low recruitment. If we take the surface water of our creek and we take pore water samples, what we see is that the pH is about a half a unit lower on average, but we also are getting acidic groundwater coming into our surface waters. We also are getting hypoxic to anoxic water. That's expected as it's coming through the sediment, but these two conditions are not good for larval oysters and for recruits. And so it suggests that the low pH at the groundwater sites might be interrupting spat settlement. So this is our spat relationship with groundwater here. This is our spat relationship with pH. Okay, and so right now my hypothesis is that we have this sort of micro layer of really, really bad water quality where we have lots of groundwater flux and the larvae either are just not settling there. I don't know if it's mortality or if they're making decisions to try to look elsewhere. So does groundwater affect populations, oyster populations in Georgia? Possibly. It certainly appears to be some response to density that we think is driven by recruitment limitation. But there are confounding variables. So if there are geologists in the room, you probably noticed some interesting things about this site that we did all this work. Again, this is what a groundwater site looks like. This is the seepage space here. One thing we'll notice, well, there's no oysters here, but this is mud. Whatever you think your experience with mud is, I guarantee you, it's not like the mud here in Georgia. We call this pluff mud. If you were to put your boat up here and step off, you're going to be in your waist immediately. It's very sinky, very soft, and we tend to have lots of it. We have lots of river discharge, so we have fairly high rates of sediment accumulation in Georgia, and so sediment is probably confounding some of our ability to detect impacts of groundwater if they exist. This is our growth experiment, and as it turns out, the bottom of the cage is buried, the oysters are partially buried, and so this is obviously going to be affecting my ability to look at things like growth rates. In fact, oysters tend to grow faster in high sediment conditions because they're trying to not be buried. The other thing that you might have noticed is that where we have this high groundwater flux is on a geomorphological feature that we call point bar. This is an accretionary bed. This bed accumulates sediment and grows outward into the creek. So this is a high sediment flux location. The oysters tend to occur on these cut banks, which are slowly migrating landward. And so we have different sediment conditions where we also have our different groundwater conditions. And so those could certainly be also playing a role in why we do or do not find oysters in this creek. So we're interested in looking at the interactions between sediments and groundwater flux using geomorphology as our sort of proxy for sedimentation. And so we select sites that have relatively high and low groundwater flux that occur on point bars and cut banks. and we looked at growth rates, and we put instead of putting the cages right on the mud where they were going to get buried, we made PVC frames, and so we kept them just above the bottom because we didn't want our oysters buried. And on point bars, the oysters grow very fast. They grow at the same sort of rate that we saw in the previous experiments. And in this high sediment condition, they're just trying to extend their shell. Again, they're trying to prevent themselves from being buried. But if we go to the cut bank sites, this is where we actually see a negative effect of groundwater. The oysters aren't growing as fast here. They're not extending their shell as fast. They don't have to, so they can put that into growing wider or putting more tissue. But once we remove sedimentation, our proxy for sedimentation, we start to see a negative effect of groundwater on oysters. And so now we see a response at the individual level of groundwater. But keep in mind that all this was just done in one single creek along the Georgia coastline. So maybe we just got lucky and this is just something that happens in Oyster Creek. And so we wanted to extend sampling at creeks that have different geomorphologies along the coastline. But we see still spatial variability in groundwater flux at all these locations, but they're all very different from each other. And when we look at all of the creeks, and we see the same patterns. So along the entire coast of Georgia, when we have oysters, the groundwater flux is significantly lower than when we don't have oysters. We see this significant negative relationship between oyster density and groundwater flux coastwide, and it's not explaining a good amount of the variability because other things are also affecting oysters. But the point is that when we have low groundwater flux, we can have lots of oysters. We don't see lots of oysters when we increase our groundwater. We also see a similar response in recruitment. The methods here are different. Logistically, we couldn't put out tiles at all those locations. And so we can only measure recruitment at sites that already had oysters because we looked at the spat that existed on those oysters. And so that's why you don't see anything below about 200. So these are recruit estimates. But again, if you squint, you might believe that there's sort of a negative relationship, but you can have lots of spat when we have low flux, and we don't really see that once we have high flux. So how might this affect management? Well, again, very few studies have examined the impacts of groundwater on oysters. And the only studies in the United States are done in Texas, where they have hypersaline estuaries, and so the groundwater there is actually buffering water quality and to the benefit of oysters. In Georgia, the groundwater really seems to be having a negative influence on oyster populations, likely due to that water quality issue, low pH. And so groundwater should be considered in management efforts and restoration site selection. The good news is you don't need fancy equipment. We have such big tides. At low tide, you can visibly see where the groundwater is coming out, and so you can very easily avoid those locations. So how does this all sort of come back to controls on oyster populations? Well, I like to go fishing. These are some of my colleagues. Some of this is actually in Shinnecock Bay. But if we want to have healthy fisheries, both commercial and sort of recreational fisheries, and if we want to be able to successfully and efficiently do aquaculture, and if we want to manage and restore and enhance these coastal habitats so that we have healthy coastal ecosystems, then we need to incorporate these new stressors. I put it in quotation marks because the relationship between oysters and sponges has been going on for an incredibly long time. Same thing with the relationship between oysters and groundwater. Neither one of those are considered in management. And so my argument is that we need to start to think about those other things if we're going to have successful restoration and enhancement of the oysters. And the question, and then you might be asking yourself why, and this is just going back to Sapelo Island. We have long-term data going back to 2005. This is just annual means. These are two sites that are just 5 kilometers apart. Well, what we can see is that we have change occurring, global changes occurring. We see salinity decreasing at these locations. We see temperature increasing over time. But importantly, it's changing at different rates. The rate of change at Hump Camp is twice as fast as the rate of change at Cabana Creek. And so in order to do effective management, we have to plan for these changing conditions. We need to be able to predict how these changing water quality conditions are going to affect all the things that I just talked about today. And so with that, I need to acknowledge a lot of people, all my grad students that have contributed to this work, and the undergrads that worked in my lab as well. This is funding resources for all the research in my lab. Not all of it is Oyster funding, but these are all my funding resources. And I will take any questions that you might have.
Chris?
Congratulations, John. That was super impressive and somebody saw you grad student.
You didn't think I'd make it here?
Yeah, I can say I'm proud of your accomplishments. And I think all of SoMAS is, so congratulations.
Thank you.
The groundwater work is super interesting, and I get the feeling, or you already alluded to it, that they, when we think about groundwater here in New York, Long Island, it's going through this super sandy aquifer, and it's rich water, and it's fully oxygenated.
Yeah.
Clearly, that's not what you have there.
Right.
It looks like it's just, is it, you didn't mention the salinity of it, but it
Yeah.
looks like it's mainly just recycled back and forth. And
Yes.
picking up the acidity and the low DO in that process, is
Right.
that correct?
Yeah, so in the surface water tables in Georgia, it's mostly just recirculating seawater that I think is when the tide is high. So we have 9-foot tides in Georgia, by the way. So when the tide is high, there's a lot of that head pressure, I think, percolating through the marsh sediment. And because that's anoxic sediment, that's also going to drive down the pH as it's flowing back through those sediments. We are on the Florida aquifer. There are in some locations freshwater sort of groundwater. But we don't have as much of it anymore because we have a lot of paper mills that have been using those wells. So along the coastline, we actually don't see as much freshwater discharge like we did historically. So that's created some problems for some of the water quality. Yeah. Yeah.
Hi, thanks for your time. I want to know if there is any relation between the location that is closest to higher river discharge and
Yeah, that's a good question. So the river discharge question. So we have a lot of rivers along the Georgia coast. And so we tend to see our oysters just along the coastline because of all that sort of water flux. But it's the fresh water, but it's also the high sediment loads. And so we don't, like if we're going up major river systems, we tend to lose oysters very quickly. But a lot of these sort of meandering creeks that may get freshwater inputs from rain events and runoff, that's where we'll see oysters. But yeah, we don't in sort of where the major river systems are, we lose them very, very quickly. Yeah. Yeah.
Hi, great talk. So as you mentioned, you observed a positive association between sponge infestation and fruit crab occurrence, like earlier on in the talk. I was just wondering if you could elaborate on this relationship and maybe provide your insights into why this might be.
Yeah, no, that's a really good question. And I've thought about it, and so there's two things that I think are two hypotheses that I have about that. One is that it's possible that the spongy oyster is giving off sort of a different cue that might be leading to attraction of the pea crabs, either as larvae or as post-set instars that are moving around trying to find a host. The other thing is, as it turns out, pea crabs will infest pretty young juvenile oysters, and I think a lot of oysters can reject them. And so the other hypothesis is that a spongy oyster might not have the energy to do the rejection, and so that might be why they have more. But those would be great student projects. But yeah, that was a good question. Thank you.
A great talk. I really appreciated how well you did there. Have you looked at aragonite saturation states in those waters?
Yeah, no, and that has come up. We haven't looked at a lot of the carbonate chemistry of the water in these studies, but I do have students that are starting to sort of look at that in lab and field surveys. I can tell you that our coastal waters are still, they're above 1, and I think they're around 2. But in the pore water, I'm sure it's probably very close to groundwaters. Yeah.
Yeah. Do you measure total alkalinity or?
Yeah. So when we submitted it the first time, that was something that we were asked, and we didn't do it in our initial surveys. But now I know better, and so we're starting to do it. we're starting to do it now, yeah. Yeah. Yeah, boss.
Great talk, John. You mentioned you presented results on the crab and on Perkensis and Apostrophidium.
Mhm.
What about?
Yeah, no, that's a good question, and we haven't tried to look at the two of them together. I know that there's been some folks that work down in Florida that looked at, I think, the distribution of the sponge and Perkensis might have been pretty similar in Apalachicola Bay, and there might have been a lot of overlap there. And we certainly can look at the data and see, but we haven't tried to explore the sponge impact on the protozoan parasites.
Position on the.
The position, yeah, the
On the
position.
title as well.
So, yeah, and so as it turns out, Jeff Byers at UGA, as it turns out, we have higher prevalence in the upper intertidal oysters than the lower intertidal oysters. And that could be sort of like a stress thing. But, historically it was thought that like the parasites, the protozoan parasites might be driving intertidal distribution, but we actually have lower prevalence in the lower intertidal oysters.
And I thought you said that Haplosporidium relationship with salinity was not as expected.
Yeah.
But I thought I saw a positive correlation in slide 42 or 43.
Yeah. So there was one with
Okay.
so there was one with temperature variance, but not sort of mean temperature conditions. So when you have more variable temperature, we have more haplosporidium. But when we look at just the mean conditions, we tend to see decrease. Now we have very warm water, and so I think a lot of the literature is from Maryland and Virginia, where I think that's why they see these increases. But we might be at the end of the thermal tolerance of Haplosporidium. Haplosporidium also probably has an intermediate host that we still haven't identified, and so we might be at the thermal tolerance for those. And I think and also we're very salty estuaries compared to most other places, which is also why I think we're maybe at the salinity tolerance for Haplosporidium where we're at as well. Yeah. Yes.
Maybe switch your topic, very interesting. Kind of sticking on this topic, I was curious in the presence of the sponge, are there any other macroparasites besides the PFAS that do digestion?
We didn't look at a lot of other things in sort of the Georgia surveys. The other macroparasites that we might find are Bunia, which is a ectoparasitic snail. I think Brad might have worked on that many years ago. And so we do see them in Georgia, but they sort of move around and aggregate. And I haven't looked to see if they're maybe aggregate at higher densities around spongy oysters or not. But I think, and then I guess we do have the Polydora, the mud blister worms. And certainly almost all the oysters have those. And so I don't know if there's really any correlation with that. Yeah. I know everybody's hungry, so thank you again for